Plants don't just absorb sunlight, they engineer it. Deep inside the chloroplast, millions of tiny antenna complexes trap light with an efficiency our best solar panels can't match. These are called light harvesting complexes, and today we will decode how they turn light into life. What are light harvesting complexes? Light harvesting complexes, or LHCs, are pigment protein complex embedded in the thylakoid membrane of chloroplast. Their primary function is simple but powerful. Capture light energy and deliver it efficiently to reaction center of PS1 and PS2. Think of reaction centers as the engine of photosynthesis, but engines can't run without fuel, right? And LHs act as a solar fuel collectors. Pigments present in LHCs. Each LHCs contain multiple pigments packed in like a network. Primary pigments are chlorophyll A, directly involved in electron transfer. Accessory pigments are chlorophyll B and carotenoids like beta-carotene, leucine, and xanthophylus. Why so many pigments? Because each pigment absorbs different wavelengths. Together, they can create a broad absorption spectrum, allowing plants to use maximum sunlight even in low light. This delivery of pigments is the reason leaves are masters at capturing lights. How LHCs capture and transfer energy? When a pigment absorbs a photon, it becomes excited. But instead of losing this energy, the pigment passes it to the next pigment molecule without electron movement. This mechanism is called resonance energy transfer or exciton transfer. Energy moves from higher energy pigments to lower energy pigments and then reaction center chlorophyll, which are P680 and P700. This funnel-like transfer ensures energy reaches the reaction center with minimum loss. Plants lose less than 10% of captured energy here. But why they are more efficient than our solar cells? LHCs consist of pigments that tightly bond proteins that stabilize them specific spatial arrangement. This arrangement is not random. It is optimized through evolution for maximum energy transfer, distance, and speed. This organization forms antenna complexes surrounding each photosystem like ring of satellite dishes. Types of LHCs. LHC2 for PS2, most abundant membrane protein on Earth. Captures majority of light energy, highly flexible. LHC1 for PS1, fewer in number but more efficient at lower wavelength. Works with P700 reaction center. Both work together to channel energy in Z scheme. Next is state transitions. State transitions is a key regulatory mechanism in photosynthesis that allows plant to balance the excitation energy between photosystem 2 and photosystem 1. Because natural light intensity is constantly changing, the two photosystems may not receive equal amount of light at all times. To maintain efficient electron flow, plants redistribute their antenna complex. Now let us discuss about state 2, PS2 overexcited. When PS2 absorbs more light than PS1, the LHC2, which is light harvesting complex 2, becomes phosphorylated, causing it to detach from PS2 and migrate toward PS1. This increases the antenna size of PS1 and helps balance energy distribution. State 1, PS1 overexcited. When PS1 receives more light than PS2, LHC2 gets dephosphorylated. As a result, it returns to PS2, increasing the antenna size of PS2. Why state transitions are important? This mechanism helps plants adjust in real time to different light conditions such as shade, sudden sunlight, or cloudy environments. By reallocating LH2, plants maintain the proper ratio of ATP and NADPH needed for downstream photosynthetic reaction. Why are LHCs essential? Without LHCs, reaction center would absorb very little light. Photosynthesis rate would drop drastically. Plants wouldn't survive under low or scattered light. LHCs make photosynthesis possible even in weak sunlight, morning hours, or deep shade. So next time you see a leaf glowing in sunlight, remember, inside it is an intelligent light harvesting network more advanced than human technology. That's the magic of light harvesting complexes. Thank you for watching. Please like, share, and subscribe.